What do this Sphingomonas bacteria and this Aspergillus fumigatus boy have in common? I'm sure you all guessed it. You can find them in extremely contaminated acid drainage in an abandoned Korean coal mine. While in this very unwelcoming environment, contaminated with various metals and other pollutants, the two organisms literally fight, giving rise to intriguing molecules called the Glionitrin.
You know, a fight like I watch on the movies. The fight, they're fighting.
Not only do these compounds look aesthetically pleasing and cute, which are of course official requirements to be featured on this channel and motivate many chemistry divas to synthesize them, but they also exhibit strong anti-tumor activity in prostate cancer cells, which is the fourth most common cancer. Moreover, they even show antibiotic activity against resistant Staphylococcus aureus bacteria, which could be very promising given we are heading straight into another pandemic threat, antimicrobial resistance or AMR. An even bigger threat to humanity, however, are people who are still not subscribed to my channel. So please do so to support me and to ensure you continue to get schooled on random topics you never knew you were interested in.
So please,
This occurrence of potentially vital natural products in very alien places is really one of the beauties of nature. The marine ecosystem has enjoyed most of the attention in the early 2000s, with many interesting compounds found in sponges, for example. We've already covered one of these compounds, namely saxitoxin, on this channel, so feel free to check this one out. Instead of going for a swim, we will go underground this video. As always, we will have some top-notch trivia and mind-blowing educating facts to start off with, and more in-depth biology and chemistry information to follow for the nerds. Many of us hope that coal mining and combustion will be a relic of the past soon, given the devastating footprint on our environment. There's definitely no shortage of headlines and scrutiny, but given coal still supplies over 1/3 of global electricity generation, it will take us a while to get there. In the meantime, some natural product chemists have found that coal mines can actually be gold mines. Acid mine drainage, or AMD, is a legit formal phenomenon which describes highly acidic, metal-rich wastewater which is formed when sulfide minerals from rocks react with water and oxygen. Bacteria actually greatly accelerate this process, leading to large concentrations of toxic metal ions getting solubilized. It doesn't take a genius to tell that this drainage is polluting and damages the quality of ecosystems. There are different ways to combat this, going from simple methods like increasing pH to more complex ones like bioremediation. But we will definitely not dive into these, as we are not sad geologists toying around with lifeless rocks. We are the organic boys and care about the weird organisms that live in the drainage and the interesting molecules that they produce. And oh man, are there many of these organisms. The myth that extreme environments are inhabitable has been debunked since the 1980s. In 2020, scientists even found microorganisms living in a 120 degrees Celsius hot rock sediment buried 1.2 kilometers below sea level in the subduction zone. This zone is basically the birthplace of earthquakes and volcanism, as it is the interface where oceanic crust is recycled into the Earth's mantle. So, if there are organisms in quite literal hellfire, it should not be surprising that we can find 1 or 2 bacteria in a slightly acidic pond. In fact, there have been over 100 studies looking at genomics of microbial and other communities in AMD, proving the richness and diversity of life that exists there. There are some key observations across the phyla, which is taxonomy that boring biologists use and that I absolutely hated to study during university. Proteobacteria, the blue ring segment, are the most dominant phylum in AMD water and impacted environments. This phylum contains some of the well-known gram-negative bacteria like Salmonella and Escherichia. Cyanobacteria, the red segment, on the other hand, are predominant in surface biofilms. This is where the light is, and they need that to do cyanobacteria stuff. Harnessing pigments such as carotenoids or also more exotic things like phycobilins for photosynthesis. However, other very interesting phyla emerge, such as the Chloroflexi. As their name suggests to today's Zoomer generation on TikTok, these bacteria are not bothered by chlorinated compounds and just flex around. Surviving in extreme conditions, they are able to engage in organohalogen respiration, such as degradation of polychlorinated aromatics like chlorinated dioxins. This is a class of highly toxic and persistent organic pollutants, such as Agent Orange used by US military during the Vietnam War. So scientists are trying to find ways to exploit these bacteria to get rid of such contaminations. Now, after we've deluded ourselves into thinking that acid mine drainage and microbes are the most important thing in the world, let's get back to our original topic, the Glionitrins. In search of untapped natural product resources, a team of clever Korean researchers took microbial samples of AMD from an abandoned coal mine and isolated various microbial strains. After co-culturing the fungus and the bacteria, so growing both species in the same medium, they observed that a new compound was formed after 8 days. This new compound was Glionitrin A. Why is this molecule special, and why did the scientists assume that it resulted from competitive interactions of the two organisms? Well, already prior to this discovery, it was known that the Aspergillus fungus produces secondary metabolites such as pseurotin A or gliotoxin. Both have distinct biochemical activities, and gliotoxin, as the name suggests, looks quite similar to glionitrin but misses the aromatic nitro group. Actually, no nitroaromatic compounds had been isolated from Aspergillus until that point. The Sphingomonas bacteria themselves do not produce any notable metabolites on their own, so it was evident that this was some sort of novel production due to co-culturing. To elucidate the biologic origin of Glionitrin A, the scientists ran a couple of experiments. First, the bacteria might drive biotransformation of fungal metabolites, such as the gliotoxin we saw earlier, to yield Glionitrin A. In one experiment, they took just the bacteria and added pure fungal extract, but without the fungus itself. And in a second one, they added pure gliotoxin, which might be a precursor to the more exotic Glionitrin A. However, no glionitrin could be detected in either setting, which meant that the bacteria itself is quite useless and does not produce the natural product. As a second hypothesis, they tested whether an unknown bacterial stimulus was altering the genetic expression of fungal metabolites. To test this, they ran a co-culture, but as soon as they could detect any glionitrin by HPLC, they essentially called a timeout and separated the bacteria and the fungus. After culturing them independently afterwards using the exactly same conditions, they did not see any glionitrin being formed. This means that just being in the bacteria's presence for limited time is not enough, and there's at least no lasting change in gene expression mediated by bacterial stimuli. Lastly, they investigated whether glionitrin could be produced just by the fungus alone, but required external stressors found in the AMD coal mine environment to induce it. Pressure testing this hypothesis basically had the researchers throw various chemicals at the fungus, such as oxidative stressors like phenol nystatin, which is an antifungal medication, as well as sulfuric acid to mimic the high acidity in AMD. This also did not lead to any production of Glionitrin A. Out of all of these results, the researchers interpreted that there must be some form of long-term competitive interaction going on between bacteria and fungus with a yet unknown mechanism. What also didn't really help was that the researchers found that keeping this co-culture for longer than 18 days led to a dramatic increase of a second closely related natural product, which they called Glionitrin B. As a key difference to Glionitrin A, this molecule is not toxic, but instead reduced cancer cell invasion, which could suggest that it might be a potential anti-metastatic agent for cancer treatment. Having completed our excursion into the merry land of microbes, we will finally talk about what we all came for: organic chemistry. Chemical synthesis of natural products is an essential tool to better understand biochemical mechanisms of action, pave the way for drug development and optimization, as well as inspire advances in fundamental synthetic methodologies. In 2021, a team at Lund University in Sweden reported the first total synthesis of Glionitrins. Let's dive into the successful approach that resulted from 4 years of hard work and many exploratory studies. Their retrosynthetic analysis started from Glionitrin B, which corresponds to ring-opened dimethylated Glionitrin A. Stepwise diastereoselective sulfur incorporation and ring closure would trace back Glionitrin A to this intermediate here, with just one quaternary sulfide group. Excluding some functionalizations that we will cover in the forward synthesis, this chiral intermediate here would be a suitable entry point for the synthesis. It would originate from an asymmetric carbon-sulfur bond formation of the corresponding triketopiperazine enolate. The researchers developed this unprecedented reaction by screening different chiral catalysts, sulfur electrophiles, and conditions. Quickly proving their hypothesis, they found that using quinine and electrophile 13A resulted in some minor enantioselectivity of the product. However, to get to synthetically useful levels of selectivity, it took countless optimization attempts and ultimately the invention of a new chiral electrophile, 13B. This reactant contains a benzylic methyl group instead of a hydrogen next to the sulfur atom. During the reaction, the methyl shows greater conformational bias because it's bigger than just the hydrogen, and thus makes one of the two transition states more energetically favored than the other. This energy difference manifests in the observed selectivity of 9:1 in the lab. To prove the utility of this new reaction, they showed that it could also be applied to various triketopiperazines, giving rise to precursors to a handful of other natural products, some of which, for example, chemotin or diphthiosylvatin, have yet to be reached by chemical synthesis. But of course, creating one or more intermediates and drawing things on a piece of paper is not the same as completing the full synthesis. If you're a proper OC nerd, you know examples for this by heart. This happened for instance during the final step in the epophylone A synthesis by Bodhi and Carrera. This natural product belongs to the class of microtubule stabilizing antimitotic agents, so it shares a common mechanism of action with Taxol. During their endeavor, a deprotection of a TBS ether, quite simple on paper, proved very challenging in reality. At any time during a synthesis, functional groups, which so far have behaved very nicely, can go rogue. In this case, decomposition of the epoxide moiety required them to explore a myriad of conditions. Let's see how the Glionitrin team navigated the rest of their synthesis, which is actually quite short and efficient. One-upping their newly established methodology, they found that the use of this inulene would result in one-pot cyclization to the triketopiperazine, assisted by the nucleophilic catalyst, and subsequent sulfonylation via the mechanism we discussed earlier. This elegantly circumvents isolation of the TKP and only required slightly increased loading of the nucleophilic catalyst at 30 mol%. It also nicely avoids use of a stoichiometric base, which would promote oxidative aromatization to the indole. The indolene can be accessed easily in 2 steps from this starting material. To further build up the Glionitrin framework, they performed a methylation, which occurred regioselectively, so not touching any other of the keto groups, as well as diastereoselectively, with attack from the phase opposite of the sulfide being preferred. They eliminated the alcohol under acidic conditions and dihydroxylated the alkene. You might notice that this 3-step sequence could have been done in 1 or 2 steps instead. For example, by using direct olefination or oxymethylation immediately, but none of these conditions worked out in this case. Afterwards, the second carbon-sulfur bond was installed through Lewis acid-mediated substitution, so elimination of the hydroxyl group assisted by the neighboring nitrogen, and subsequent addition of the thiol nucleophile. Fortunately for them, this afforded the isomer that they needed. This was the kinetically preferred product, because under acidic aqueous conditions, it actually turned out to equilibrate to the thermodynamically favored isomer with opposite configuration. Finally, they just had to deprotect the thioethers and create a central disulfide bridge. Deprotection with boron tribromide gave the dithiol, which was oxidatively closed by using a disulfide reagent. This completed the synthesis of Glionitrin A, and to get to its brother, Glionitrin B, you just have to cleave the disulfide bond again and methylate the 2 thiols. For some funny reason, the conditions that were reported by the Korean team that originally isolated and recreated Glionitrin B in the lab did not work at all. So they had to use a different way, which was another thiol redox dance and dimethylation, proceeding in extremely high yield. Ultimately, their synthesis required just 8 or 9 steps, with 15% overall yield from the abundant starting material. Also, the entire lab work takes around 7 days, and if you remember the introduction, it required co-culturing of at least 18 days to form any glionitrin B at all. This makes a strong case for chemical synthesis, because we need less than half of the time and can engineer structural analogues that can further elucidate glionitrin's activity and mechanism of action. This wraps up today's video, and I look forward to seeing you in my next one. If you are new to the channel, you can check out my other videos, and if you're already a true follower, you can always rewatch them again and flame me in the comments for my bad audio quality. All the best, and until next time.